Thank you very much for inviting me, giving this seminar. Can you hear me? Yeah. Because this microphone is only for, yeah, okay. So yeah, it's a pleasure for me to come back to Seville. Always a pleasure. So I'm working on the impacts of climate change on forestry mainly. And today I decided to talk about the niche, a concept that will soon turn 100 years old. Because 50 years after Hutchinson gave its definition that revolutionized biogeography and ecology, I think it's time to push the concept a bit further if we want to address the huge challenges climate change poses to biogeography and species distribution modeling especially. Because as you know, species distributions are changing. Many studies have shown poleward shift or upward shifts, mainly in animals, fewer in plants which are sessile organisms. But still, even in forestry, there are some studies showing, for example, like this one showing here, this is hallmark, expansion at the southern range of the species expansions, but usually for forestry it's difficult to disentangle the effects of climate change with other effects like management, because, for example, hallmark in these regions has always been disadvantaged in the management in favor of other oak species which are used for timber. But still, for other species like herbaceous species, we see clearly upward shifts, so shifts towards the summit of the mountains. This is another study using data, French data, on the French flora data, showing that species have moved towards the summit on average 60 meters in the last decades. And even in forestry, more lately, some studies have compared the distribution of seedlings to the distribution of adults across elevation gradients. And they have shown that for most of them, seedlings were distributed at a higher elevation than their parents. So if you look at all the studies which shows shifts in the distributions, you will mainly see changes like this, that is, shifts towards higher latitude or higher elevations, either just of all the range or just the northern margin, or sometimes it's just the southern margin moving, or sometimes only the optimum of the distribution of the species shifting towards the pole or upward. You have a few species showing the reverse trends. And also you can have species expanding northward and southward or retracting. And why do we see so much variation in species responses? This is first because climate change is, even it's a global change, it varies across the planet. The changes are, for example, much higher. The increase in temperature is much higher at higher latitudes than in lower latitudes. So the climate change is not the same everywhere on the planet. And then species sensitivity to this change is also can be very, very different. And the species' ability to adapt to this change, either through phenotypic plasticity or microevolution and migration, is also very different from a species to another. And above all that, if you take also in account of land use change, landscape fragmentation, biotic interactions, pollution, etc., you can imagine how fuzzy the picture can be and how different the situation for different species can be. So if we want to be able to predict species distribution, future distribution, what do we need to know? According to Hutchinson and later according to Soberon, the distribution of the species, it's the geographical representation of its realized niche, which is the interaction between its fundamental niche, that is the abiotic conditions in which a species can persist, with the biotic conditions in which a species can persist, that is all interactions either positive or negative with other species, and also the ability of these species to disperse, to migrate, and also we need to take account for barriers to dispersal. So let's come back a few minutes to the fundamental niche, because obviously if our ability to describe the fundamental niche is too poor, there's no chance we're going to get the right pictures of the future species distributions. So from my point of view, Hutchinson really made a revolution when he proposed this definition of the fundamental niche more than 50 years ago at Cold Spring Harbor Symposium on Quantitative Ecology as the set of environmental conditions allowing individuals of a species to survive and reproduce indefinitely. It was really a big turn for biogeography, modern biogeography and ecology, because Hutchinson's view of the niche is an hypervolume of n dimension. And so you can describe the niche according to several dimensions, which are actually the environmental variables. And the species performance varies across the range of each of these variables. It shows usually an optimum at the center of the range of each variable. And if you are able to describe how the species performance varies across these different gradients, you can model the species fundamental niche. And so this definition of the fundamental niche by Hutchinson had the premise of the first species distribution models that appeared a few decades later. But when we want to describe this fundamental niche, usually what we have only, it's not usually species performance, it's usually species presence or absence, like in these maps, where you have occurrence of different subspecies. And for each location, you can get a description of the environment. Usually it's climate variables or soil variables. And so you can describe with this variable the presence of the species, in which condition it is able to survive. Okay, but usually when the species is absent, we don't know. I mean, when it is absent for another reason than the climate or the soil, you can bias your estimation of the fundamental niche, because in this fundamental niche you don't consider other variables than climate or soil or other abiotic conditions. If the species is absent for another reason, for example, due to an interaction with another species, you will bias your estimation of the fundamental niche. So anyway, this definition of the fundamental niche gave birth several decades after to the first species distribution models. So I will use the term SDM for species distribution models. Well, they are called now correlative models, but at that time they were called envelope model, referring to the Hutchinson hypervolume. So basically those models, they use occurrence data of a species and information of the environment, and they, using different statistical techniques, they relate this information to those ones to explain why the species is occurring here and not here. So this is the first correlative models that had been published in the early 19th by Everson and Prasad. As you can see, you see here why they were called envelope models. So basically, they were describing using forest inventories. In this case, it's poplar, trimming poplar in US. And so they were describing, according to different environmental variables, in which range of those variables the species were occurring. And by putting all that together, they were able to predict. These are the data used. These are the prediction of the simple model, of the envelope model. So these models have been widely used to predict species distribution, either in present condition, in past conditions, in future conditions. They are by far the most used to predict species distribution, even though they raise large amount of technical problems. But I think this is not really the question now. I think the question is really, we have to go back to this definition of the fundamental niche and push it further. Why? I think the Hutchinson fundamental niche allows very well to describing the species distribution, but not really understanding this distribution. This is not because, you know, the species can live in this range of temperature that it will explain you why it's living, why this is the optimal range of temperature for this species. Also, Hutchinson fundamental niche is a static view of the niche, and it supposes that species distribution are in equilibrium with climate. But now they are no more in equilibrium with climate. And also it supposes that species niche cannot evolve, but we know it does. We have, there's been many papers the last 5 years showing that niche is actually evolving, and very quickly in some organisms. For example, this is an example on mosquitoes showing a critical trait here, which is a critical photoperiod to get into diapause. And the population that was screened over this latitudinal gradient here for this trait in control condition in the lab, this data show that this critical photoperiod within 5 years has decreased of about 1 hour, meaning that now those mosquitoes are entering diapause later in the season than before. Okay, so because they can take advantage of better condition in autumn to do one more generation. And even in organisms which have longer generation time, because mosquitoes is doing many generation within just 1 year. In other organisms like salmon, which have a 2-year generation time, we can see evolution, genetic evolution going on due to climate change. This is data on pig salmon in Alaska, and the trait as the authors have looked at is date of migration back to the cold spring where those salmons reproduce. And they show that within 40 years, this date has been earlier by 2 weeks. In the meantime, temperature, as you can see, has increased by more than 1.5 degrees Celsius. And when they have monitored those salmon's population for decades, actually, since the early 70s. In the early 70s, they had some genetic variation within those population with earlier migrant and later migrant salmons. Okay, and now in the population you have only almost only early migrants, and they got the neat idea in the 70s to use a natural genetic marker to follow the late migration alleles, and from this data, from this natural marker, they were able to show that late migration marker allele has decreased very sharply in a few decades in those populations. So here we have a case where the heritability of the traits under selection is quite high, 0.5. We have a strong selective pressure. Temperature has increased by 1.5. The generation time is quite low, 2 years old. And so in a few decades, key traits for the niche of this species has evolved, was able to evolve. So I think, well, from the beginning of my research, I've been looking for another definition of the niche that would allow us to go further with species distribution modeling. And quite early, I thought we should consider the fundamental niche as a set of phenotypes that allow individuals of a species to survive and reproduce indefinitely in a set of environmental conditions. I think this definition, first, it fits better to the theory of ecological niche, because it's much more easy than to explain character displacement and niche segregation, concepts that were already there, defined by Hutchinson 50 years ago. And also I find it's better suited to study niche evolution, as obviously niche does evolve, and it is better suited to account for species adaptation to climate change in our models. And more specifically, to take account for phenotypic plasticity and to microevolution of the traits which are key in the fundamental niche. So actually, this definition has been preceded by a second type of species distribution models that we call process-based models. And this is a kind of model I'm developing in my group for 3 species. This model, which is called PHENOFIT, this is a contraction of phenology and fitness, because this is a model that makes the hypothesis that the occurrence of key events like leaf unfolding and flowering, fruit maturation, or leaf senescence, the occurrence of these events along the year relatively to the occurrence of some stresses like drought and frost are key to understand the survival and the reproductive success of the tree at each location where you can find it. So to give a bit more details about this model, you have to simulate precisely the phenological events, the exact date of leaf unfolding, flowering, fruit maturation, and leaf senescence. And then you have to see, so you do that with phenological process-based models. You also described resistance to stresses with other process-based models, resistance to frost and resistance to drought. And then you can, when you aggregate all this, you can get an estimation of the annual survival probability of the species and its reproductive success, and so get an estimate of the fitness of the tree. So this model uses daily climatic data and data on each of those traits, because you need to model those traits and to get to know the reaction norms of each trait to each environmental variable. This is a kind of results you can get with this model. This is Cecil Oak. This is the observed distribution, and this is simulated distribution. So in this case, this data have not been used to drive this map. We use them only to estimate the performance of the model. Okay, and then of course we've been using them with climate scenarios to forecast species distribution changes. Now there's a few process-based models, even for animals. Just to mention that, this model is called Niche Mapper. It has been developed by Michael Kearney and colleagues. And it's just to say that even for animals, you can develop such models. In this case, so this is a model for Greta glider, a possum, and what the models predict is grams of babies produced each year. And so for that, you need information on basically so many things: the body shape, the body mass, the thermal conductance of the fledge, the diet, the efficiency of the milk to produce grams of babies, etc. And they succeeded to simulate the distribution of these species quite well, and they were targeting also a region where for now we have no record of the species, but the species could live in those regions. So just to say that you can do such models also for animals. Now you have models also for insects, for birds, but you need quite a lot of data. So I say that these kinds of models were able to take account for phenotypic plasticity, because you describe some traits that usually are plastic, phenology or resistance to stresses on plastic traits. This is just an example to give you an idea of how we can do that. Basically, this is, for example, for leaf unfolding date or flowering date. You know that for tree species, birds need to break a dormancy, and for that they need cold temperature, and then when dormancy is broken, you need warmer temperatures for the cell growth, basically. And the reaction norms to temperature during this period and this period are different. But you can, if you have, you can measure some of the, you can measure the parameters of those reaction norms in the lab. It's not really easy, but you can do that, or you can infer them statistically using data series, time series, or different data spatially distributed. And so those reaction norms, since they are reaction norms of traits under selection, you usually find local adaptation for those reaction norms. And if you have data on several populations, you can derive the reaction norms of the different populations to take this local adaptation into account. So, with these kinds of models, we can address some questions that we cannot with correlative SDMs, and especially I will just talk about two of those questions today. The first one is concerns phenotypic plasticity. We can investigate how much of the species range or the species niche the phenotypic plasticity explain, that is in current condition and also in future condition. So we try to do that with the PHENOFIT model. So we did that for 3 species. Actually, not this one, but 3 species. So I just recall you that we can describe and we can get the reaction norms of different populations. So first of all, sorry, we did that for one trait, for leaf unfolding date in this study, okay? And this is just to show you how leaf unfolding date varies across years because of the reaction norm to temperature, because climate conditions are not the same from one year to another. This date varies, but it varies as well between sites, and this variation between sites is either due to, it's both due to phenotypic plasticity and maybe also to local adaptation, but at least to phenotypic plasticity. If you make transplantation, for example, of some trees living here, you won't see the same leaf unfolding date because of the phenotypic plasticity, okay? So what we did, we made an exercise with the model. We compare different situations, different models. Model 0 is the model where the leaf unfolding date varies in space and time due to the reaction norms. So it varies between years and across the range. And then we had a model where we fixed the date. We removed the plasticity. We removed the part of the plasticity that explain interannual variation, okay? So we did that by comparing model 0 to model 1, where at each location, the dates are fixed to the local average and do not vary between years. Then model 2, we removed the effect of plasticity on spatial variation by fixing for each location the date to the average date over the range. Okay, so dates do not vary anymore between years and do not vary across sites. And then model 3, where we fix the date to the historical date and to the future date. If you use a model with climate scenarios, you will get other dates because of the warming. And then you can have the effect of the impact of the plasticity on the trend you get in the dates due to the trend in the warming. Okay? So comparing these 4 different models, so PHENOFIT, but with plastic or fixed date, you can disentangle the year-to-year variation from the spatial variation, from the trend variation of in the dates. So get the different effect of plasticity. So we did that on those 3 species, beech, Cecil oak, and Scots pine. So some keys to understand the results, which are not always easy to understand. This is a fitness map you get with plastic dates. So the dates are changing every year at each site and are changing across the sites. Okay, that's the normal case. This is a fitness map when you have fixed the date, either to the local average or to the range average, the historic one or the future one. This is the occurrence data. And this is the difference between those maps. So showing in red, plasticity is advantageous. In blue, plasticity is disadvantageous. Okay? And the figures you will see on those maps is the overall advantage all over the range. Okay? So first of all, when we compare model 0 to model 1, we get the impact of plasticity, the impact of the interannual variation of the dates due to plasticity. And what we see is that this impact is low, 2%, 7%, 3%. Okay? It is much lower than the effect of the plasticity on the spatial variation. Then you can see, and so this difference is because the variation of climate and of photo period across the range is much wider than the variation you get at each location across years. Okay? Then we can see that for beech and oak, plasticity is advantageous at the warmer margin and disadvantageous at the colder margins. And this is because when you fix the date to the range average, what you do is you fix here dates which are later than the date that the normal date you will get because of the plasticity, which is an earlier date. And this earlier date allows the trees to finish their maturation. The probability of success of the fruit maturation is higher. And reversely, at the northern margin, if you remove the plasticity and you fix the date to the range average, you have a date which is later than the date that would occur due to plasticity. And so it decreases the probability of fruit maturation. As you can see, Scots pines show very different pictures than the two deciduous species. And we have a very high load of plasticity at the southern margin. And this is actually due to a developmental constraint because Scots pine needs much lower temperature to break the dormancy of the buds than the two other species. And basically, it cannot live out, it cannot birds cannot burst because they are lacking cold temperature to break the dormancy. Okay? So it's a developmental constraint that we see here. That was for current conditions. Now for future climatic conditions, the picture is quite different. As you can see, the impact of plasticity on the species range is much higher. For the two deciduous species, it's higher but negative for Scots pine. So for Scots pine, this is because all over this range, because of a lack of chilling temperature, Scots pines cannot live, cannot make new leaves actually. And so he's disappearing from this region because of this developmental constraint. And for the two deciduous species, plasticity helps the species cope with climate warming. As you see, the plasticity is advantageous even at the core of the distribution because it helps the species following climate warming. So the take-home messages of this study are that phenological plasticity is not always adaptive, and it can translate developmental constraints. So plasticity has some limits. We see that in Scots pine. The year-to-year variation in phenological dates have a weak impact on the fitness, except at the margins. And because trees have long-distance gene flow, we believe this suggests that plasticity in trees, in forest trees, is selected as a scale, a wide scale, maybe the scale of the species range. And finally, phenological plasticity will be advantageous in future climate conditions for most species, increasing their range compared to no plasticity. Okay, the second issue I wanted to talk about concerns microevolution, because this is another issue correlative SDM cannot really address. Especially two real open questions at the moment are, can microevolution slow down the extinctions we see in the simulations, or can it accelerate the colonization of newly suitable habitats at the poleward margins, because dispersal traits are also able to evolve quickly in such conditions. And such questions call other questions like, are currently populations at their phenotypic optimum? Which provinces will perform better in future conditions? And what are the selection gradients across the distribution currently, and how do those gradients vary with climate, and how will they vary in the future because of climate change? So those are questions we are trying to address now. What I will show are preliminary results. So what we are trying to do with PHENOFIT is to estimate selection gradients. So what we do basically is that we fix the date of leaf unfolding to different values that are observed across the species range. This is a distribution of the leaf unfolding date, or the species distribution. So we fix the date, and then we can calculate the average fitness of the species across the range. And this is what you see here. This is the mean fitness over Europe of the species, depending on the leaf unfolding date. So as you can see, there is an optimal leaf unfolding date, which is approximately the average date we see currently in the populations. And apart from this optimal date, the fitness decreases, but it decreases much sharply for later dates than for earlier dates. And this is because the later the leaf unfolding date, the shorter the growing season for the tree is, and therefore the lower the probability of fruit maturation is. So the load on the fitness is much higher. And here, the decrease in fitness is due to spring frost damage, because if the leaf unfolding date is too early, it is susceptible to frost damage. But it does not impair fitness as much as if the growing season length is too short. So what we see is asymmetrical stabilizing selection on this trait. And if we look at the fitness landscape at each location over the range, you will see, so this is different fitness curves for 3 locations. You can see that it varies across space, and actually it varies across time too. This is a selection gradient you can estimate at each location across the range. So red is selection for later dates, and blue selection for earlier dates. And you can do that for in the future. If you're using climate scenarios, you can estimate the selection gradients for the future. And if you have information on the heritability of the trait, you're looking at the genetic variability within the populations, then you can estimate how fast this trait will be able to evolve due to climate change. So that's what we are doing at the moment. This is a work in collaboration with Ophélie Ronce, Étienne Klein, and Sylvie Muratorio. We are comparing the selection gradient we estimate with the models with selection gradients we can estimate in the wild, in the populations. And we are also trying to estimate the relative contribution of plasticity, of microevolution, and of gene flow. And we are coupling the PHENOFIT model to a quantitative genetic model and a demographic model to be able to take account for genetic evolution in our simulations for the next centuries, because now we have access to climate forecasts for several centuries, and we can also use the model in the past during the last millennium and see how genetic evolution of those traits because of climate change the picture, the whole picture. And I'm going to stop here, and thank you for your attention, and thanks my collaborators.